So thank you for the kind introduction, and thank you for the invitation to present our work and for putting together this very nice meeting. I guess I will point on the left screen for me. So today I will want to focus on data center approach that we developed in the lab, and still, of course, studying the mitotic spindle. So this work was mainly done by Jan Lequinf, an assistant professor at that time in the lab, as well as a bit of Benjamin Merca. And at the end, I will also present work from Nina Soler and Maciej da Silva, who are two former or present PhD students in the lab. So we are interested in the cell division without a lot of surprise. You will be convinced already that it's an important step in the cell. And our underlying question, almost philosophical question, is how something that looks for a physicist a bit sloppy could be that faithful. So the estimation that I can find was 1 error over 100,000 divisions. And to do that, we will further focus on the metaphase, the time where all the chromosomes are congressed in the middle and connected to the mitotic spindle by microtubules. So the mitotic spindle, if we think about it, there is a lot of work on the biochemistry of the mitotic spindle. There is aspects of structure, but what we will be interested in today is really the dynamics and the mechanics. And these aspects have many folds. There is the molecular motor that are, of course, in their activity dependent on the load that resists to their action. There is the elasticity of the microtubule that can bend and resist. Dynamics of these fibers, obviously, the cross-linking, which can make microtubules slide or simply passively cross-link them. All that piled up together make it a complex system in the meaning of physics, meaning that you cannot really expect to study each component and superimpose each component to get the working of the total system. And with this idea, we wonder whether we can have a data approach, say, well, let's forget about all the hypotheses and say we look at the data and wonder what is happening. So we do that on the nematode Caenorhabditis elegans, which is a classic system for that. You already had a glimpse this morning about it. It's a non-pathogenic worm of 1 millimeter. We're interested in the first division of the zygotic division. You have here the microtubules that are labeled and the chromosome. And what I would like you to consider on this picture is that it's an asymmetric division. So there is stronger pulling forces on the posterior pole than the anterior, and I will need that a bit later. If I have a more magnified view, you have here a 3D view of the chromosome and the spindle poles. And I would like you to appreciate that there is a large metaphasic plate, and then there's probably a wall in the viscous drag about it. I will also come back to that. Just before moving to the deep part of it, we have various classification of the microtubule. It was already evoked this morning as 12 microtubules that transmit the force from the periphery to the poles. The microtubule of the spindle, either emanating from the poles or either branch from other microtubule, here in purple. And we have kinetochore microtubule in orange that are connected to the chromosome. The C. elegans system has a peculiarity. It's holocentric chromosome, so we have connection dispersed all along the chromosomes. Now, you probably remember already this picture from Steffi Edeman this morning. Most of the microtubules connected to the kinetochore are not reaching to the pole. And this is an important aspect to consider because you cannot really assume the half spindle as a kind of a solid or gel-like system. And this will come back several times. Now, how to approach in a data fashion the spindle. We use the spindle length. It's a well-established proxy for the studying of the spindle. You have here various proteins disturbed in this publication. And you see that they are all on the kinetochore, not really in the spindle itself. And you see that you have various signatures on the elongation of the spindle, so the length between the two poles. And so we'll use this length as a proxy. And just before starting, I would like you to consider the aspect of variability because I will need this idea. In a genetically identical system, so the people that did the study used this plasmid with two fluorescent reporters, one green, one red, and put it in bacteria. And when you have bacteria transfected with such a plasmid, you would maybe imagine that you have yellow bacteria everywhere. In fact, you have a variegated colors, which suggests that even on the same plasmid, the same quantity of the plasmid, there is variation in the expression. And so we will be back to that because we ask about question about the variability of the spindle elongation in the C. elegans. So that will be our first item for today. The second one is looking at the small variation of the length, still on a data fashion, and see whether we can deduce some insight into the spindle mechanics. And at the end, with the two ideas that come out of this aspect, we start wondering about the mechanism by looking at the dynamics. So let's start by the first one. So to keep it simple, we have a tracking of the centrosome. Because they spread on many pixels, we can have an accuracy much better than the classic diffraction limit. And if I look carefully at the spindle elongation over time, I see that there is a shallow elongation during metaphase and then the classic elongation during anaphase. If I zoom in the trajectory, I see that there is in light green, the high frequency noise, fine. But there is also this modulation in dark green that contains information about the mechanism that connects the two poles. So we put that in a data system. I will detail in just a moment, but before that, I would like to tell you a bit more about the data set. When we do a data center approach, a key aspect is, of course, the data that you put in. So we gather many experiments of the lab done in different projects with a bit less than 80 gene depletion or mutation, including kinesin, microtubule binding protein, subunit of dynein. We had also repeats of non-treated and control embryo, varying the temperature, varying the labeling. And we put that together and wonder what is happening. So if we plot the average trajectory per condition at the same temperature, 18 degrees, we see that we have a large variability of this elongation. This was somewhat expected. We have in black the average curve. But now let's do a second observation and get only the non-treated embryo. It doesn't look really less, it's not really much more condensed or much more reproducible. We have here the average of the non-treated embryo. So there is also this probably an aspect of intrinsic variability, even if they are all genetically identical. So we wanted to get an understanding of that, and to do that, we put 4 objectives: reduce the complexity, because comparing just curve of elongation is not really very convenient. Have a representation that is interpretable, but we want to have a pure data approach. So no hypothesis based on the previous literature and no feature that could be based on the previous interpretations. So we tried different dimensionality reduction approaches, and we ended up by using principal component analysis. So for the one that doesn't know that, it's a way of observing the variability. So you suppress the average curve, the black curve that I showed before, and you wonder how you can change the axis. If you imagine that you have 2 dimensions, to a first axis along the direction where the variability is the largest, a second orthogonal axis where it's a bit lower, and so on and so forth. So here for 2 dimensions, it looks, of course, trivial. When you have many dimensions, because you want to compare curves, it makes the things a bit more complicated. So here you have a typical reconstruction with this approach. I use a clasp 2 RNAi, so a protein that is attached at the kinetochore and regulate the dynamics of the microtubule. And you have here the noisy curve is the experimental curve. In green, you have the reconstruction with 1 component, in red with 2 components, and in blue with 3 components. And so it looks that with 3, it works quite well. Of course, we cannot do that on waving, and there's a proper way of doing that. We can plot a curve representing the number of components that we put in the reconstruction, and on the y-axis, the average reconstruction error. And with that, we see that we have a classic L curve, and in the corner, we have the optimality. So 3 components, and with that, we can account for 95% of the variability. In fact, each component accounts for 70, 19, and 6%. So once we had that, I made a kind of graphical heuristic equation, let's put it like that, to recapitulate the fact that the elongation pattern we see it as the average combined with archetype, which is this 3 component extracted from the PCA, and a certain dosage, so a single number that say how much of each of these archetypes you are putting in. Now let's look at this archetype because they are really informative about what is happening in the spindle. So the first archetype looks to be flat, and it's probably encoding roughly for the average length of the spindle. So it's quite related to already known feature done manually. The second one in a range is an elongation both in metaphase and anaphase, quite well known. And the third one is much more surprising. So we find this shortening of the spindle. So, of course, combined to the others, we don't see any shortening each time. But there is this component present and needed to explain the variability. So now, what we can do with that? The first way we did with a data approach is looking at which gene perturbation have a high coefficient or very low coefficient for this in front of each of these archetypes. So for the first component, very high, we have protein from the central spindle, so you have the feeling that the spindle is breaking at the time of anaphase, so that's why it comes with a high length. And for the very low, you have TPX2 homologue, which disturb the length of the spindle. It's known in the C. elegans. For the second component, we have once again protein that lead to a breaking of the spindle, the clasp of the Zen4, which is a kinesin 6 in the central spindle. And for the one that fade to elongate, you see here in orange, they are really flat. Then you have ORB temperature sensitive, but at permissive temperature, the important part is separase or the lack of cortical forces that contribute to elongate the spindle in C. elegans. And for the third one, we have, so again, the TPX2 and the clasp, which is not really surprising because we expect defect in the kinetochore, because the community knows that this shape of shortening is usually related to defect in kinetochores. But with very low third component, we have for now something that I'm not able to explain. We find 2 genes that have no non-direct effect on the spindle, but make worms that are shorter on their life length. So the spindle is a bit more squeezed, maybe, I don't know. It could be a matter of discussion. So if I now come back to the third archetype, then I took the CLS2, so one of these examples with a very high coefficient in front of this third archetype. We see this shortening, definitely. And so in this one, you have either defect in the attachment or either breaking of the spindle. It's known that there is kinetochore defect. In fact, I will not detail all the conditions, but other conditions with defect in the kinetochore attachment also come with this kind of phenotype. Now, from here, we can wonder, well, maybe this third archetype is just because we included some condition with defect on the kinetochore attachment. So what we did is different approaches, because of course the data that we provide to the PCA are key to decide the archetype that we extract. So we use only 500 embryo and repeat 500 times this taking of a subset, sampling a subset, and extracting the PCA coefficient. And we plot it each time, and here you have in a gray shading the standard deviation around the archetype in this bootstrap. And we see that, in fact, there is not really that much variation of this archetype, which means that it's not really just a few embryos that are key in deciding this archetype. Now, of course, in the 500, we have a mixture of perturbed condition and non-treated condition. So we push the thing a bit further and consider only the non-treated embryo. These are the plain lines in this diagram, and compared to the whole dataset archetype in the dashed line. And we see that once again, it's not really that change, which means that it's not only the few conditions with a kinetochore defect that decide of these archetypes and decide of this third archetype. This third archetype is already present in the non-treated embryo, even if maybe not weighted that much. We also wonder about the temperature, but because we reduce the number of embryo per condition, we have archetypes that are a bit more noisy, and changing the temperature We have also a change in the pace of the division. That's why they look shifted on the right for the one at the lower temperature. But nevertheless, we keep roughly the same shape, which means that it's not dependent on temperature. It's not non-treated versus treated. These archetypes appear to be robust. And there's another idea if we push a bit the thinking forward, which means that the difference between the treated and the non-treated is not a kind of change, a complete change. It's just the dosage of the same archetype, which means that there's a kind of continuous change between the two. And I will come back at the end on this idea. So now let's have a second look at these archetypes. And there's a second aspect that was striking us. If we look at these archetypes, they are all three across metaphase and anaphase. We have not specialized archetype on metaphase, specialized on anaphase, which means that if I give a dosage of the three archetypes, it should apply to both phases. So in other words, we ask the question, maybe what is happening in metaphase is already deciding what will happen at the end of the anaphase, even if what was reported was that the central spindle is not really focusing like in other cells. It appeared that there is no microtubule at the very early anaphase in between the two chromosome sets, and then there is an assembly of the central spindle. So what we did to keep with a data approach, we formed a small convolutional neural network, and we put a bit of the trajectory around the anaphase onset. It's here highlighted in purple. And the expected result is the final length. We trained this network on 950 embryos. And so we did the training, validation set, and we tested on about 500 embryos. And when we do that, what we obtain is that the predicted lengths fit very well the real measure lengths on the test set, which suggests that we are able to predict the final length with only this small portion of the curve at the time of anaphase onset. We wonder whether it's important to have it at this right time. So we assume we name the Pearson coefficient the predictive power, and we plot it over time by moving this window where we took the data, and we see that we have really a steep increase of the predictive power when we start putting data at the very end of metaphase, early anaphase. Now we ask a last question about that and say, maybe the PCA coefficients are enough to recapitulate this information that allow to predict the final length. So we did another PCA extraction, but this time only with the bits of trajectory -5, 20 seconds before after anaphase onset. Because if we put the whole trajectory, we are providing already the information in a way or another. And with these three coefficients, we can once again predict the final length only with the three coefficients on this very narrow part of the trajectory across anaphase onset. So with that, I would like to conclude this first part on the keeping an eye on the time on this data approach. So we find that there is only three components. So let's think a bit about that. It means that there is maybe not really that much independent process happening in the spindle, because we have only three coefficients that are able to represent this elongation pattern. And in other words, it limits the possibility for the pattern. We cannot, with three numbers, we cannot really make an absolute crazy variations of the elongation pattern. The two first archetypes, so the archetype are robust, I guess that was clear for you. The two first ones are more or less overlapping the manual feature that were used previously. So it's detailed in the publication if you are curious, so we can talk about that later. The third archetype about the shortening was a bit more puzzling for us because it's already present in non-treated. And we were wondering what it could be. Is it maybe the signature of a mechanism that corrects the attachment or something like that? I don't know. There is this part of the central spindle, which has this length determined already in the late metaphase, very early anaphase, even if it's assembled de novo. We have this non-treated versus perturbed embryo, and this is an idea that I like because it suggests that the phenotype, it's not all or nothing. We can have a continuous description. And with this idea in mind, we say maybe we can use now the PCA coefficient as a kind of predictive tool to say, well, there's a gene that I don't know. I extract the PCA coefficient and look where it maps with the others. And that's exactly what we did. So you have here a diagram. You have various conditions. The detail is not really important for now. One color per condition. And you see that if you put each embryo, so you have the coefficient of each archetype. And they are clustering, which means that the same condition produce roughly the same PCA coefficients. And now you can, of course, imagine that you can map an unknown genes and see which gene targeting is next to it, and maybe make a statement or experiment based on this insight. I will stop now for this approach for the sake of time, because I would like to show you another approach of the spindle length that we did a bit earlier in the time, in reality, looking at the small variation of the spindle length over time. I will just give a quick look at that. Before that, I would like to clarify an aspect, because I very often have the question. I will tell you about the maintenance of the spindle length over the duration of the metaphase, not the length that we have at the assembly of the spindle. It's probably a different process. And it's well known that when we go through the first divisions of the C. elegans, then the length of the spindle is scaling with the cell size. You can have also the same idea represented with curve a long time. So this was published, and it's not really our focus today. What we are interested in is this shallow elongation a long time. So what we wonder is whether there is a mechanism that could maintain the length. If you read the literature, it's assumed that there is a mechanism that maintains the length during metaphase. When, as a physicist, I'm a physicist by training, so I'm not very convinced of a mechanism that maintains the length when I look at this shallow elongation. So what we did, no worry for the ones that are not familiar with the data stuff. What we did is basically focusing on the small fluctuation, the small variation of length. We did that by multiplying by a hand window, because there is a drift to get rid of that, and doing discrete Fourier transform, average over embryo. We use a data model. I will just come back to that in a second. Convert it with the hand window in the Fourier space, and we did the fitting with some care to be sure that we have no problem with that. And about the model, of course, there is plenty of model of spindle length. So we cannot really decide for one model or the other. So we say, well, maybe we should rather use a data model, so something that is simple, that fit well the data, that looks reasonable from what is known for the spindle. Of course, I can derive this model from the spindle, but I can also derive some others with different assumptions. So I prefer to really say that it's a data model. The fitting looks nice. So we have reports because we have the windowing of the model, the convolution with the hand window. That's why we have that. So the black dots are the experimental data, and the maroon curve is the fit. I should have said something a bit earlier. What we did is, instead of taking the whole curve of the elongation, we make a 15-second window, and we move it by a step of 5 seconds to represent each moment of the spindle. Because, of course, if I do that for the whole spindle, I could have an equivalent mechanism, but this mechanism is probably useless. There is no mechanism that's probably governing the early time of metaphase, late metaphase, early anaphase, late anaphase. There is so much things that change. So we rather focus on small time periods. And doing this kind of fit at each of these times, we can extract the coefficient of our data model, so stiffness, viscous drag, and inertia over time. And when we do that, we find something really surprising. The stiffness is really low compared to the two other coefficients. So we can compute the instantaneous power stored or dissipated in each of these components. And we find, without a large surprise, that the one in the viscous drag and the inertia is really larger, which suggests that there is probably no mechanism that maintains the length of the spindle, just slowly elongating against viscosity and inertia. So I will stop here for this part because I just wanted to bring you the idea of this spindle spectral signature. There is, of course, much in this project, but I have no time to dig into it. And the fact that there is no maintenance of the spindle length. And at that time, if I put that together with the idea of the first project of this shallow elongation, we were wondering, so there is probably something that we are not really very clear in the mechanism of the spindle. And so we wanted to have a look at that and look at the dynamics of the microtubule within the spindle. And this will be my last part. So the dynamics of the microtubule in the spindle, the most prominent phenomenon is the poleward flux. So the poleward flux is the historical experiment by Tim Mitchison. So they photoconverted tubulin in the spindle, and they saw that this tubulin appeared to move to the pores. So I have here, okay, my pointer is not really working very well. Anyway, so you have here in dashed line the position of the wheel dimer of tubulin, and they are moving towards the pole. But of course, because there is dynamicity of the microtubule, the microtubule looks apparently the same. It's a bit like a river. If it's not flowing in a quiet fashion, you have the feeling that the river is not moving, but there is permanently water entering on one side and getting out on the other side. So there is various mechanisms that were proposed for this poleward flux. The most classic one is probably the thread milling, where you depolymerize the microtubule at the pores, and in a coordinated fashion, you polymerize it at the kinetochore, so on the chromosome side. So it was reproduced in vitro in a minimal system, to be sure that we understand it very well, by the lab of Maria Zan. The second mechanism that were proposed were based on the sliding of overlapping microtubule. You can also make the same argument with bridging fibers. And by cross-linking the kinetochore fibers, then you can pull on them and carry them with you. And the last mechanism that can work is you have chromokinesin that bring the arms of the chromosome in the middle to have a thin metaphasic plate. And so by reaction, you are pushing on the microtubule towards the pole. In all these mechanisms, the idea is that the half spindle would move as a gel towards the pole, so all the microtubule at the same rate. And this is the thing that we will interrogate in a few seconds. So we did that in the C. elegans at a high frame rate, 10 Hertz. That's why it's not, because the dynamics of the microtubule is very fast. That's why it's not very clear. And so to make the things clearer, we took the median pixel by pixel of 10 embryos registered on the bleach area. And you see here the bleaching and the recovery. We wanted to quantify that. So we took the fluorescence profile along the spindle axis. We can pile up this profile over time, and we have a kymograph. In this kymograph, you see the red lines, which correspond to the very bright centrosomes. You see a dark line in the middle, which is the shadow of the chromosome, where there is no microtubule. And you see here, yeah, I'm not sure my pointer is really helping you a lot. Here in the middle, I guess I will do like that. Here you have a part that is closing, which is the bleach area. So the surprising part was that it closed with a kind of V shape, which was not at all expected. We were expecting closing the stuff with no flux, as previously reported once, but with a very low weight of imaging at that time, or moving on one side, on the other side. So we quantified that, and I will not detail today, but it's in the bioarchive, and I can discuss about that. On the side of the chromosome, there is no antipoleward flux. It's just the dynamics of the microtubule that is growing with the diffraction of the microscope that make this pseudo moving, which is, can say, artifactual if you want. But there is a larger slope on the kinetochore side, which suggests that there is an additional mechanism. So for this additional mechanism, we wonder what it could be. So we tried different genetic perturbations that I will not show today. And one more physical experiment, I would say, without genetic perturbation, where we bleach close to the chromosome or far to the chromosome and measure once again the shape of the closing. We still have the V shape, and we have larger speed when we are close to the chromosomes compared to the case where we are far from the chromosome. the two rightmost columns. And so from there, we consider that maybe there is only the kinetochore microtubule that are undergoing flux and the spindle microtubule are not moving. You remember that the kinetochore microtubule are, for most of them, too short to reach the pole. So you can imagine that they slide along the spindle microtubule. And indeed, their speed is dependent on the dynamics of the microtubule, on their attachment to the kinetochore. So that fits. So now we wonder how we can do that. We will need to slide parallel microtubule. For now, we have not included any kind of mixture of polarity. So we assume that we need to have parallel microtubule sliding. So there is a kinesin that can do that. That was reported, that was a study in vitro. So the counterpart in human cell, of course, KIF15. And how you slide parallel microtubule. Either you assume that the motor, the tetrameric motor, is not working at the same speed when you have an head with the tail in the back and a head with the tail in the front, and that's why you have a different, or you accumulate at the plus end of one microtubule and keep working on the other one. So we have an homologue of this protein, which is called CLIP18 in C. elegans. It's needed to focus the meiotic spindle. Whether it's just doing cross-linking or sliding the microtubule actively, I don't know. And it's nicely present in the mitotic spindle at metaphase and in the central spindle at anaphase. So it could well do the job. So we try to target this protein. So our first try was by RNAi, but because we need it for the meiosis, we can have only a mild RNAi. So we have a little decrease of the slope, but was not really crazy. So we tried a different strategy using a temperature-sensitive mutant. So there's plenty of them in the C. elegans world. And at restrictive temperature, when we use the mutant, we have really a lower slope compared to the control condition. To be complete, I must say that we have the same at the permissive temperature, either because the protein is already not working correctly or any other consideration. You will also notice that there is a huge change of the slope between the two temperatures, even in the control condition, but this is known. This is the dynamics of the microtubule that is changing with temperature. But with that, we suggested that the CLIP18 is maybe one of the motors that could make the job. So to conclude this last part, I would say that we have kinetochore microtubule that would slide along the spindle microtubule, which break with this idea of a gel-like half spindle. It's something that is much more flexible, probably with the CLIP18, but maybe some additional mechanism, and we have in mind dynein adapted by Numa on the minus end of the kinetochore microtubule. And with that, I would like to bring you a few reflections on the perspective that we are doing. What this mechanism could be good for. So if we have a motor in between, this motor, of course, will have a dynamics in working and in detaching that is depending on the force. So maybe it's a way to insulate the kinetochore from what is happening on the poles. And in this division, which is asymmetric, you need to pull hard on the pole to orient the spindle and position it. And you don't want to report this force directly, transmit this force directly to the kinetochore. So maybe the motor will have a role. And the idea is, of course, that you let maybe the microtubule slide, and even if the length of the spindle is not maintained, what we saw, it may be not really important. It's maybe more important to ensure regulated forces on the kinetochore. So we are in a nice system where the forces on the two poles are different. So we measure the flux on both sides, anterior and posterior. Anterior, there is a lower force, and we saw that the flux was different. We were not expecting it in this direction. We're expecting it the other way. So for today, I have no explanation on this result. And at least if we suppress the forces by targeting GPR12, we rather equalize the force, but with a higher level. So for now, we don't know the mechanism of it. So to try to dig into that, we made a minimal system, but in the shape of an agent-based simulation with cytosine, putting only the essential components, so the kinetochore microtubule nucleated at chromosome, the spindle microtubule that are attached to the pole and that are dynamic but not undergoing flux, the KLP18 motors, and the spindle microtubule can push on the chromosomes. And with that, we can reproduce the flux, but the simulation is not yet really stable in parameters, so we need to have still work on that. So I will finish by a few perspectives. So at some point, we wonder whether this stuff is a kind of a peculiarity of the nematode. But when you look at works in the HeLa cells, in Dan's lab or in Eva Tolich's lab, they also saw that the flux is decreasing with the distance, which is not really consistent with the idea of a gel-like spindle. So maybe there is something happening also in human cell, even if there is probably many differences as well. So we saw that there is no length maintenance, and this is also this shortening. And maybe this different aspect by the data is telling us about some mechanism that regulate the force, or maybe that even correct defect in attachment, because we have our holocentric system have about 30 microtubules per chromosome, 25 to 30, which probably prone to a merotelic attachment, and there is correction mechanism. So are we seeing that? I don't know, but we are wondering. And now I will finish by telling you that for that, probably the elegans is a convenient system because it's much less complicated than the human cell, I have the feeling, because we have no depolymerization of the microtubule at the pole. So we have fixed spindle microtubule. So we are looking only at the kinetochore microtubule moving. It's probably a much more comfortable situation for this kind of data or modeling approaches than a full complexity mammalian spindle. With that, I would like to end by thanking all the people in my lab. The one that contributed mostly in the work are in the bold front. Our collaborator in the Mathematics Institute in Rennes, Loïc Lemarec, collaborator in Toulouse, Sylvie Tournier and Yannick Gachet, people that provided us with trains, various people with whom we discuss science and progress in our understanding. And of course, all the agency that funded these studies. With that, I would like to thank for your attention, and I would be happy to answer questions.
We have time for a few questions.
Yeah. So you were talking about how you believe that the non-kinetochore microtubules aren't moving; those are static. I could imagine that if they were moving, that might be difficult to measure because they turn over so rapidly. So I was curious, how confident are you that you think they really aren't moving, or do you think maybe that they could be moving and just be hard to measure?
So if they are moving slower than the others, that's for sure, because when we measure the flux far away, it's slower. The point is we targeted the MCAC, the KLP7, which is the classic way of saying the thread milling, and we have no effect on the flux, which is not really very consistent with the idea of depolymerizing the spindle microtubule at the poles. So we have it's not impossible all the time in biology, but it's lower if there is a motion of the spindle microtubule. And for now, we have no indication that it could be. We have other indication in the other way. We also wonder about the statmin and the fidgetin or CLIP13, which is a kinesin 8 homologue, to do the job of depolymerizing, but none of them were explaining the stuff. No change in the flux. So it's already difficult to show that something is not existing.
Hey? I have a question on the asymmetry that arises in the C. elegans.
Yeah.
So when you did your experiments to bleach, does one half spindle behave differently from the other?
Yeah, the flux is different. The length of the spindle is almost the same.
Yeah.
And in EM picture, there's not really much difference. So that's why we rather think of a functional difference in the dynamics of the component rather than a real structural difference between the two half spindles.
Yeah, so the spindle that's probably pulling towards the cortex is the one that behaves in a way that the kinetochore microtubules are the only ones that flux. Am I right? Or both sides?
I guess the kinetochore are the only one to flex on both sides, probably. That's because we don't see, we see the same pattern of decreasing when we bleach close or far on both sides.
On both sides, okay.
But the point is the flux is not the same on both sides. And which also was a weird question, because at the beginning, that's why I draw your attention on the large chromosome plate at the beginning, because we saw that we are pulling on the pole. This force is transmitted to the kinetochore in the classic gel-like model. There is this plate, we transmit it to the, so can you imagine that there is same forces all along the spindle. And what it says that it's maybe not. Maybe there is sliding, maybe this large metaphasic plate is opposing a drag force when it's moved towards the posterior. And so it makes an imbalance in force between the two sides, plus sliding in the spindle.
I had a question.
Yes?
So you nicely argued that there's this continuum of responses that the PCA reveals, but I was curious, have you thought about predicting like the limit of this continuum, where like, I would imagine if you add enough dosage, like the whole machinery fails. This was sort of the continuum of still the spindle separates, but is there sort of a limit where if you add enough of this treatment, the spindle fails to separate?
We have tried. At the same time, we have a condition that are quite stringent where the spindle is breaking.
Yeah.
And even in this condition, it's still explained with the same archetype. So we have not found a way of breaking the fact. Maybe it's possible, but.
Ooh, all right, thank you again. Oh, maybe one.
Right, so the three archetypes that you obtained from the PCA analysis.
Yeah.
Will that percentage persist through the lineage of division?
We have not tried the next division. So I don't know. Would imagine that it will probably stay more or less the same, but it's a pure speculation. I have no data to support that.
With that, let's thank him again. Thanks.